Dear students, myself Dr. Jitendra Kumar, Assistant Professor in Biomedical Department, BVRT Narsapur. Today we are going to see the introduction of tissue engineering. It's a professional elective fifth which is in 4th year, first sem. So let's start the introduction. First we'll see the course objectives of this course. So this is first is to provide a general introduction to the use of artificial material in human body for the purpose of adding healing, correcting deformities and restoring the lost function. And another objective of this course is it is expected that the student will have successfully completed the elementary courses in the mechanics of deformed bodies and introductory course in material prior this course. So let's see what is tissue engineering. So tissue engineering is a biomedical engineering discipline that uses combination of cell engineering, material and suitable biochemical factors. That biochemical factors can be media and growth factors, some growth factors to restore, maintain, improve and replacement the different type of biological tissues. Now, why tissue engineering is important? Because as we can see here that there are thousands of organs required, but we are not able to supply those organs. So we'll try to make it in a lab. So we'll try to regenerate that tissues so that we can fulfill the requirement. So here we can see like these are the organ requirement and these are the organ available. So from here we can understand like requirement is more than the supply. So we'll try to make it at lab and we'll try to regenerate those tissues and organs. Next steps of tissue engineering. So from that pictures we can see first step is cell isolation. So first task we do cell isolation. We took cell from like a patient or a human being. Then we did expansion of that cell, means we do the culturing of that cell. Then we do this enhancement or proliferation of that cell, and then we prepare 3D scaffold. This is the scaffold which is used to provide these physical or mechanical support to the tissues. And then after we did the tissue development by adding some cells and some growth factors and maybe some media and other things, chemicals. And after then we'll after making this tissue we'll implement to the patients. So these are the overall steps of the tissue engineering. So cell isolation, expansion, 3D scaffold development, then tissue development, and then the implantation to the patients. Now, scope of the tissue engineering, as we know, like tissue engineering is very emerging field, so it has various applications and scopes. So here we have mentioned some of those. Like first is organ and tissue regeneration. So by it is very important in developing the artificial organ and tissues such as skin, cartilage, bone, and other like heart and liver. And next is cell therapy. By utilizing stem cells we can regenerate those tissues which are got damaged, and we can implant to the patients. Next scope is 3D printing and bioprinting. So creating complex scaffold structure that can mimic the bone structures or extracellular matrix, and which can support the bone in growth. Ultimately our goal is to grow the cells, and hence we require that bone in growth or tissue growth. Next is drug delivery system. So this is the another scope of tissue engineering. So it is the delivery mechanism within the scaffold and localize and control that release of the drugs. And next is a disease modeling and organ on a chip, developing microphysical system and engineering tissue that can be replicate the human organ. Now in this course we are going to cover, these are the topics which we are going to cover in this course. So here first unit will discuss the basic principle of tissue engineering, and then we'll see details of the bioreactor, what are the bioreactor, how it functions. Then we see the scaffold fabrication, we'll see what is a scaffold and what are the various techniques to fabricate, and what kind of software or what procedure or process we'll use for fabrication will be discussed. Then we'll see the applications of that tissue engineering in engineering and transportation. Then we talk about the clinical trial in tissue engineering. So these are all are covered during this course. So now course outcome after completing this course, what student will be able to explain the growth and organization of the dynamic of the cell, and the student will be able to explain the quantitative aspects of tissue engineering, and then will be able to explain the approaches to transplanting engineering cells and tissues. And next, the student will be able to tell us about the organization and modeling of tissue engineer. Then student will be able to apply the tissue engineering concept in various organ regeneration like liver, kidney and others. And next the student will have concept of the clinical trial in a tissue engineering. So these are the outcomes after completing this course a student will be able. Thank you. That's all. See you in the classroom. Thank you.
Like, share and subscribe. Hit the bell icon for more updates.